aeruginosa. It is a small gram-negative aerobic bacilli possessing a single polar flagellum, which allows them to the mobility. Um, some associated pathologies are respiratory and urinary infections, hospital infection, nosocomial, and septicemia are present in patients undergoing prolonged treatment with immunosuppressants. Main virulence factors are biofilm formation ability and composition of the extracellular matrix of biofilms. The, the three main curum sexing, cysteines, flagellants, Verdin, therefore as an iron-based system, type 4 pili, lipopolysaccharide, and other membrane proteins. The type 3 and 6 and 2 secrete system and relates to the extracellular milieu, lytic enzymes, and elastase.
Rhamnolipids are surface-active glycolipids. They are amphipathic molecules with an unique nature and have a complex chemical structure. They are composed of two moieties. The first and the hydrophilic portion consists of a L-rhamnose sugar, and the hydrophobic moiety consists of a hydroxydecanoic acid, linked by a glycosidic bond. They could have one or two sugar or fatty acid molecules, so they can be classified as monorhamnolipids or dirhamnolipids. These variations made rhamnolipids one of the most versatile natural amphiphiles and give them their surfactant activity, meaning that they reduce the water surface tension significantly, making them great for emulsions. Amongst all the virulence factors of P. aeruginosa, rhamnolipids are involved in bacterial adherence, biofilm formation, and its maintenance, which is particularly helpful in respiratory infections establishment. Rhamnolipids typically are not considered a significant member amongst all the other virulence factors of P. aeruginosa that cause disease, and this is because they are not the main conducive factor of the immune response against this bacteria. They can act in many ways, but most of them are focused on maintaining favorable growth environment. This is endorsed by the fact that the liberation of rhamnolipids is highly regulated by the three main quorum sensing systems of P. aeruginosa, which are RHL system, LAS system, and PQS system, Pseudomonas quinolone signal, which job is to link the actions of RHL and LAS. In fact, one of the actions that rhamnolipids do is raising PQS molecules' solubility in water, thus enhancing cell-to-cell communication, improving the bacteria's stress response against damaging conditions like oxidative stress and antimicrobial agents. Also, its condition as a surfactant make it very efficient to disperse biofilms. This is important because in the face of adverse conditions like environmental changes, such as fluctuation in oxygen levels, alteration of nutrients, or increasing of the toxic products in the tissue, P. aeruginosa cells will need to leave the biofilm to colonize another less threatening part of the tissue. Furthermore, this dispersion capability can also affect the biofilms formed by another bacteria. Another mechanism by which rhamnolipids enhance the bacterial growth is the formation of vesicles. It has been seen that they associate to form micelles that serve as carriers for self-produced toxic compounds, which they deliver to other bacterial cells, enhancing and accelerating their elimination. They also have a role against the immune response. They can induce histamine release from mast cells, stimulate degeneration and release of the inflammatory mediators from human platelets, and principally they induce the lysis of polymorphonuclear leukocytes. Also, they inhibit cellular function and produce damage to the bronchial epithelium by affecting the tight junctions structure. P. aeruginosa causes serious infections in immunocompromised patients. Because it's an opportunistic bacteria, it has the facility to infect almost any part of the body, from the skin to the lungs and even the gastrointestinal tract. Between the most serious diseases that can cause are pneumonia, septic shock, urinary tract and gastrointestinal infections, even skin and soft tissues infections. And rhamnolipids' role is to provide an environment free from immune cells, other bacteria, or toxic substances that can affect the bacterial growth or its biofilms.
According to some estimates, microbial biofilms are involved in 65% of infectious diseases and more than 80% of chronic infections. Then, 65 to 80% of total human infections are associated with biofilms formation and include periodontitis or dental caries, cystic fibrosis lung infection, chronic otitis media, infective endocarditis, chronic prostatitis, chronic wounds, recurrent urinary tract infection (UTIs), bloodstream infection (BSIs), ventilator-associated pneumonia, and infection associated with indwelling medical devices. The prevalence of HCAI is estimated to be between 5.7% and 19.1%. In the United States, it's estimated that the mortality rate due to HCAI is around 5.8%. The use of silver salts and silver-coated seams have been one of the methods to prevent adhesions and therefore infection. This technique has been useful as it reduces the inflammation generated by the biofilm or delay the infection. However, it is not capable of preventing or stopping it for a long period of time. In the same way, antiseptics do not retain the infection in catheters, so they are not definitive solution. The industrial systems and the medical devices industry currently relies on mechanical cleaning and oxidative biocides. The former removes biofilms and later gradually dissolves the biofilm matrix material and eventually kills the sessile cells. Intervention strategies currently used for biofilm control will either prevent initial device contamination, minimize initial microbial cell attachment to the device, penetrate the biofilm matrix, and kill the biofilm-associated cell. To remove the device, the use of 0.05% sodium metabisulfite added to catheter acts as an intravenous antiseptic and eliminated left atrial colonization and endocarditis. Other ways is the use of silver salt and silver-coated seam.
In this case, we propose the use of rhamnolipids as antibiofilm agents in the prevention of biofilm-associated infections on hospital surface, such as catheters and another medical device. The objective of these proposed are to make an effective product to prevent and eliminate the development of biofilms. Another is to use rhamnolipids as antibiofilm agents to dissolve the biofilm matrix. And the last is to reduce the incidence of infectious disease associated with them, improving public health. The detailed description of the way the virulent factor is going to act to dissolve the problem. The solution of the biofilm matrix, the rhamnolipids interact with the biofilm matrix to dissolve in the structure and allow the penetration of antimicrobial agents. The other is eliminate bacterial cells through the antibacterial activity of rhamnolipids. And the other is prevention of a biofilm formation, inhibit the adhesion of bacteria to surface.
Material and reagents. Pseudomonas aeruginosus strain, THI agar medium, minimal M9P specific medium for biosurfactants, incubator, centrifugal, spectrophotometer, microscope, rhamnolipids of Pseudomonas aeruginosa, crystal violet strain for biofilm measurement, Cyto 9 biofilm viability measurement, solution saline, dilute samples and maintain sterility, solvents, ethanol and ethynyl acetate, purify and store rhamnolipids, buffer. Bacteriology lab, Petri dishes, Bunsen burner, HPLC, chromatography columns, catheters, autoclave, and other medical equipment.
Objectives are evaluate the ability of rhamnolipids to inhibit the formation of biofilms on catheters of surface and another medical device. 2. Determine the optimal concentration of rhamnolipids necessary to prevent the formation of biofilms. 3. Analyze the antibacterial activity of rhamnolipids against bacterial strains common in hospital infections. Points of methods are the production of rhamnolipids, cultivate Pseudomonas aeruginosa in half LB for 24 hours, transfer the strain to amine medium supplement with glucose, 20 grams per liter, and incubate for 48 hours. The supernant and purify the rhamnolipids for high efficacy liquid chromatography or similar methods. 2. Evaluation of antibiofilm activity. Prepare surface of catheters and another sterilized medical device. Inoculate bacterial strains, Pseudomonas aeruginosa, Staphylococcus aureus, Escherichia coli on surface. Treat surface with rhamnolipids concentration of 0, 1, 5, and 10 milligrams liters for 24 hours. Evaluate the formation of biofilms using violet glass or cytonine staining techniques.
Follows the analysis of bacterial activity, where Starbury cultivate bacterial strains in LBO TSA medium for 24 hours. Then treat the bacterial cells with rhamnolipids, concentrations of 0, 1, 5, 10 milligrams per liter for 24 hours. Assess bacterial size viability using colony counting techniques. In addition, the following is performed: evaluation of stability and toxicity. Evaluate the stability of rhamnolipids under different storage and use conditions. Determine the toxicity of rhamnolipids in human cells using cytotoxicity assays. Statistical analysis. Results will be analyzed using appropriate statistical tests, ANAVA, students, etc., to determine of the results.